Hi, everyone. Hi, Dr. Nabil. How are you doing?
Hello. I'm great. Hi, everyone.
Awesome. So, in this session, we will be talking again about personalized medicine as we did the last time. And we will mainly focus on cancer because there will be many sessions, as we say already the last time. But this time, we'll fight cancer smarter, how we use AI to revolutionize the personalized medicine, and especially, as I said again, in cancer. So, welcome and let's start. Okay, I think you see my slides perfectly, right? So,
Perfect. Can see it.
okay. So, yeah, this is one of the last session actually, or the resume of the last session. We have already talked about how genome is unique. This I really like this sentence. Each genome is unique and makes each person is unique, right? So how the personalized medicine treats each person individually because each person is unique and has a unique genome. So we have already talked about how the all I mean one treatment cannot fit to all persons, and we have to tailor the treatment upon the person because each person is unique. And yeah, so we talk already about what is omic data, what is non-omic data, and clinical data, and how we can assemble all these data to make this network and to make a perfect treatment or personalized treatment for the person. So this is mainly the last session. If you didn't see the last session, please refer to it and then come to this next session because I think it makes a lot of sense. Yeah, so since we will be talking about cancer, just a brief introduction about cancer. I mean, we have the expert here, but still, I'm working also with the cancer data, so I may present a very short introduction. Mainly cancer, we know cancer. Cancer is a disease, obviously, with thousands of
Yeah.
diseases. Actually, it's not one disease, it's 1,000 disease in one. And basically, we know it's uncontrolled cell growth. So it happens when the cells start to divide quickly. It's mainly like a machine when it has on and off, and all of a sudden the off fades, right? So the machines keep going and going and going. And yeah, so the cancer could spread, obviously. I mean, it could go into nearby tissue, which we call metastasis. And as I said, it's not one disease, it's hundreds or thousands of diseases, and there are hundreds type of cancer. Although the cancer might be, would say something like random, but in itself is not random. And that's what we'll see in the next slide, how the cancer could escape the immune system, for instance. So here on the left side, we see a normal cell division, which has a certain systematic way to do, and it is controlled by a series of number of proteins and how the cancer cells divide. So obviously to survive, how the cancer is surviving or how the cells are surviving, it's not random. It's really inside a very straight, or I would say controlled way that these cells are growing. And also they use a certain system to grow, and this system is really in order. I mean, it's not random, it is ordered system. And that's why we hear often this microenvironment, because the cancer itself is forming some microenvironment inside the environment, and this environment obviously is our body, and this microenvironment, this is these tumor cells where they start to grow. And yeah, they use a different strategy, which I find myself very fascinating. I mean, the immune system, how they escape the immune system, they escape this checkpoint. Basically, it's like a police. Our body is under control by a police, and this is police when we say this is immune system, especially the T cells, and they are presenting their ID to T cells, and they say, hey, I belong to this country. And basically the cancer cells should not belong to this country, I would say, and so should not belong to this system, and usually they should be captured by the T cells. But in case of cancer, they are not catch because they skip there. They do some camouflage, right? And camouflage that's they skip this system. So we have also some they become invisible to T cells. They can also invisible to macrophage, right? So they resist the cell death. We have cells which usually they are under or they are continuously growing, right? Continuously growing. That means there is a grow and there is a death, there is or there is a born and there is a death, and so on and so on. But the cancer cell actually it's immortal. I mean, it does not die, and basically because it escapes this cell death, which is the normal way of living cells. Then it has also a controlled proliferation, obviously, because it suppresses some proteins, and also it activates other suppressor usually, or other they are suppressed, they are activated, so vice versa. And they also use some sustained angiogenesis. It's really smart because they use some blood vessels, so they construct a new blood vessels where they bring nutrient and oxygen to survive. They also cause genome instability, and I think this is well known and by many biologists. That's because the cancer cell itself is has some variant. It is as long as it grows has a much more variant. So the variants are introduced quickly, and these variants they are given always some advantages. So yeah, it causes really genomic instability. So it does also some metabolic reprogramming, means that usually the cell is using oxygen, and usually we use glycolysis because there is a fault of oxygen. However, the cell or the cancer cell is using directly the glycolysis, which is a very smart way also because it learns how to live in very harsh environment, and it could survive also
Yeah.
in low oxygen. And
Yeah.
yeah,
Yeah.
it has ability to invade, and as I say, to grow quickly and to invade other organ, then easily to stay. So yeah, we have the expert here. You
That's,
can add.
no, I think
Go
it's
ahead.
it's great you have mentioned the most important, way how cancer survive. So you use several methodologies to survive. As you already mentioned, that cancer is a multifactorial disease that is characterized by the uncontrolled, what we call cell proliferation, which is resulting from the dysregulation of normal cellular processes. You are mentioning here quite a few cellular or normal cellular processes like, um, immune system vigilance, basically can escape from cell death and proliferation, cell arrest, all this. It's using a system to grow and to resist to treatment. So as you said already, the cancer is a clever and can use different methodologies and processes to grow and invade other areas, and also can escape from programmed cell death. And also by escaping the cell cycle arrest can also induce genomic instability, and that could also lead to the more mutations and more resistance. More mutation means also more resistance. And I think that slide mentioned the most important biological processes, how cancer could escape from the control and could survive basically to treatment. Yeah,
Yeah,
excellent.
absolutely. Yeah. Um, so yeah, what are the cause of cancer? I think we talked already about this in the last session, but nevertheless, I mean, it's important always to remember or recall or beside again these causes. So we know already that there is a part of inheritance, obviously, and this inheritance maybe it's roughly between 5 to 10%, right? Where it can occur because it's inherited from the parents. However, we know that most of the cancer cause is during the life span or the life or coming from the lifestyle of the person. So basically, I think first cause is smoking, and there is obesity, there is this diet or poor diet, lack of physical activity, pollution, and so on. Yeah, there are different factors. Obviously, also we can cite much more, because depending also where it is, if it is in some poor country or developed countries. So there are many different risk factor.
That's true.
Obviously, yeah, you can add to this.
Yeah,
Yeah,
I think here this slide summarized very well the cause of cancer. You already mentioned only small portion of inherited or acquired mutations that could lead to cancer. Of course, in this 5 to 10%, there are some mutations that are inherited, but most of the mutations that are acquired during the lifetime, what we call the somatic mutations, and that represents more than 90%. And basically, this 5 to 10% of genetic alterations represents only 5 to 10%, and more than 90% is mainly due to the environmental factors. As you already mentioned, there are many factors there. You highlight very well the most important risk factors like smoking, obesity, infections, lack of physical activity, pollution. And all of those actually impact gene expression. And you can see basically gene expression can be controlled by epigenetics, but also by genetics. And both of them, they are communicating. Either the risk factors can come from the inherited, what we call the genetic alteration, inherited or acquired, but also can come from the epigenetic mechanism alterations. So that's basically the
absolutely.
connection between the both causes of cancer.
Both causes. Yeah, indeed. So here, since you already cite epigenetic, yes, here we show also the epigenetic mechanism. I mean, we will not go into detail, obviously, but we know that this is most of the mechanism of the factor that they are causing cancer. So basically, when we have a DNA methylation, or for instance, a histone methylation, or a non-coding RNA, for instance, this microRNA. So yeah, they are different
Yeah,
different mechanism, but the result is the same, is gene dysregulation. Because most of the thing when we talk about gene, what is variant? So if there is no impact on the RNA and then a protein, they basically we will not see. So if we have, for instance, there are some variants which are in introns, we will
exactly.
not see the impact on that. But if there is an impact in the transcription, then the likely or the possibility to have an impact in protein could be high. Yeah, so this is mainly.
Yeah,
Do you want to
yeah.
add something to this slide?
No, exactly. I mean, that's what you have
Yeah,
mentioned. That's correct. Basically, those are the major epigenetic mechanisms. Basically, what they are doing is to express more genes or silence genes or sometimes switch off and on genes. And this gene expression regulation, which can silence some really key genes like proto-oncogenes, some tumor suppressor genes, but also can overexpress some oncogenes, and that's the case of cancer. So cancer mainly is more oncogenes, proto-oncogenes, and less tumor suppressor genes. And epigenetic can regulate
absolutely.
those oncogenes and tumor suppressor genes. And basically by switching on and off those key features that control the uncontrolled proliferation. That's what I want to add here. Thanks.
Yeah, indeed. Thank you. So yeah, this is a very, I would say, it's summary here about the global threat and of the cancer, how it is alarming, how the cause of death. So in 2020 we have nearly 10 millions of death. This is quite a lot. I mean, this is
Yes.
it is a global threat that's require
Yeah.
actually. As we mentioned here, urgent action, and it's not only that. We show later how this is ongoing for the next years. So yeah, most common types we know that's breast cancer, lung, colon, rectum, and prostate. Since they are the most common, though, it might make sense to prioritize the attention to this type of cancer, obviously. And we have risk factor. We have already cited some on the last slide, like tobacco, this BMI or BM index, alcohol, poor diet, and physical activity. Yes, and cancer prevention is very important. It requires addressing these factors to make the cause less, obviously. As we say, that's oncogenic. Here, this is another point, very important. It's really depend on the low-income country or low-income country, where we find that most of the cause of cancer could be infections. And this is really also alarming because, then it makes also a big difference between countries and the type of cancer of country, which makes it even more complex to make, again, the treatment adequate and so on. That means each country or each group of country could be treated differently because the causes are different and the type of cancer is different. So curability, so many cases can be created with very early detection. And this is mainly I would say this is the most important. This is the most important point. If you predict it and you can prevent it, then this is much more important or much more efficient than detect it and find the treatment. So this is what is scary, I would say. This by 2030 would be 20% raise of the cancer death, and by 2050 it will be 70%. This is really scary number. So we are not talking about something which is 2 or 3%. We are talking about 70%, and we're not talking about 100 years. We are talking about 20 years or 25 years. It's not that much. So this is a really urgent to work in. The research should
No, this is scary figures and alarming figures, and one should really expect that science, there's a lot of innovation, there's a lot of progress, and while we are still all those cases growing every year, and then you can see 20% by 2030, 75 by 2050. But when you look to the table that we have there, a lot of type of those major cancer or type of cancer can be prevented. And you look to risk factors like tobacco, high BMI, alcohol, poor diet, physical inactivity, and even infections. So if you tackle all of those, probably you could reduce massively the incidence. So that the major thing. I think we're looking for more innovation and identifying the new technologies that can detect early cancer and treat better the cancer. But also we need to educate people to avoid all of those risk factors, because those risk factors, they are modifiable. You can modify them. So basically we have to educate people to be aware those are the major factors that induce cancer. So that's what I want to add here.
I totally agree. I mean, awareness from childhood, not from later age, or from the childhood, where we have to emphasize probably everything related to these risks and so on that people can prevent it. Right. Let's go a bit into the topic, or more into the topic. So here I emphasize here a bit about the difference between cancer, why cancer again is very complex. And so we have two factors here, probably more than two factors. So the first one is the type of cancer and which organ the cancer is affecting. And inside each organ, we have also a different type of cancer. So this is really vast, and also it's like branches, right? And we have the second factor, which is also another important factor, which is a gender specific, right? So we cannot say, for instance, prostate cancer in case of women, and can neither say uterine cancer or ovarian cancer or cervical cancer in case of male. So here again, this is an addition factor, addition complexity to the cancer research or cancer disease itself, which again show this diversity or genetic diversity, how it could be so vast, and how again this research should be expanded not only to narrow one type, but instead to see the overview about everything.
That's true. I mean, the gender specific cancer is one bigger issue. Of course, you already mentioned the cancer heterogeneity, the inter and intra-tumoral heterogeneity, very important. So within the same tumor, you can kill the 70%, 80%, but always there are some cells that can resist because not all the tumor cells they are the same. So there is a changes in the mutations, and we know also the cancer cells that are mutated rapidly and can evolve and basically can resist
Yeah,
to treatment. So there are many cases there. But of course, one of the most complicated one is the difference between male and female. Already mentioned some type of cancers. They are much more associated with male and others with female, which make it complicated to have similar treatments for both genders.
yeah, absolutely. So understanding the gender specific is also another very
Yeah.
important point. Yeah, again, here, just the traditional and modern approach. I think we talked already about this the last session, but never mind. Here again, how the traditional method here or approaches are, like, for instance, there is a chemotherapy, there is a radiation, and there is also the side effect, right? So, however, if we make that very personalized or treatment tailored to the genetic makeup of the tumor, then we can avoid this genetic the side effect. Yeah, so mainly this is how it is personalized medicine, how we that's make the future of medicine very very different and very interesting at the same time.
Sure.
Yeah, when we talk about personalized medicine, we can not talk about the AI, obviously, although this is not a very new topic in medicine, but still, I mean, the technology is going very fast, and we have to incorporate each new technology into it. Yeah, basically, we have a massive data already, but we'll have much more massive data. So just an example about cancer sample. When we have a cancer sample, usually we have a sample, and we have a tumor sample, we have a normal sample, and just two samples, they can make already 1 terabyte. Imagine, just without analysis, we're not talking about analysis. So imagine how the data is massive, and this is adding to that the complexity of the data, obviously. Adding to that the fast we should be analyzing the data, and because the one patient cannot wait till 3 4 years to say, okay, I will develop an algorithm coming to your bed. No, a patient needs his data or the output very quickly. So this is the fast analysis, and the AI learns from thousands of patient data. This is a really, really important a point here, which non-human being would be able to do it, and neither and no classical algorithm would be able to do it except with the deep learning. Yeah. Anything to add?
Yeah, I think those point there really important. Why we need AI, because will make our life easy, dealing with the big data. But one of the most important thing is what we call the hidden features in the AI for cancer. Sometimes we are guided by our knowledge as a biologist or as a scientist, and then we sometimes we tend to forget about hidden or what we call features that are not visible or predefined by us by scientists or by human, but can be learned by the model through data training. And this is very important because, as we already discussed, when you treat the patient tumors, you can kill 80%, 90%, but always there are some small populations, they have different features that could resist to treatment. So if you are able to identify those small population with those key features, you could target them. And
Yeah.
this AI for hidden features, it could be for omics and non-omics. What I mean by omics and non-omics, so the non-omics, anything which is like imaging, like MRI, PET scan, CT scan, but the omics mainly for molecular aspects. So you can capture the non-obvious relationship between the what we call omics and non-omics, and can improve the predictions and reduce the noise. So that's very important when we are talking about precision medicine, and not to just treat what is obvious, but we want to treat what is hidden also. And the AI will allow
Absolutely.
us to treat the hidden features and hidden populations, which can grow again as recurrent, and then can show some resistance. Yeah, that's what I want to add there.
Absolutely. Yeah,
Yeah,
absolutely. I mean, this is a very, very vast topic. We can talk
true,
about that, and
true.
also not only talk about that's what's already present in the publications. It's an ongoing creative process. I mean, you can always think about any area, immunology, or how the cell, for instance, what we say about the cells, they are hidden from the human system. It's there, it's already a word, right? An ocean of information, and there you can target any part of the pathway to catch or to make a new treatment. Or yeah, there is a lot of things to do. And
Yeah,
basically here I just cite some how can AI do in cancer. And basically it can, since we have this early and accurate diagnosis. And for instance, we can already from the MRI, from the CT, from the X-ray and so on, we can already detect tumors, right? We can already use this data to detect a tumor, and hopefully and very early stage. Personalized treatment plans. Obviously, we talked already about that, but here again, how can we plan that from the genomic data? Again, everything what we use right now for to treat this data at genomic level, it's use deep learning, because basically there is nothing now we can. It's because of the big data first, and also because we still have a lot of noise. So because of the noise, to reduce this noise with this background, we need a very accurate data to be trained and to give also a very accurate result. Because we don't have to forget that a trained set or subset of the data we have to train should be really high quality. This is something people might miss, but this is a very important point. And the drug discovery and this and development, obviously we can always, if we have already an idea about the treatment, we can already simulate and make models to predict some or to discover some drugs. For the prediction is the same. So if we can make, for example, we talk about that later probably, but we can also make some models to simulate the data, right? And the data, I mean, here you have already the drug for the patient and how this patient will react to this drug. So here we can also make some in silico data, and we can check how we predict the outcome. Workflow. I think workflow automation is very, very important
true.
point. Again, this is to make everything systematic, right? And to make everything like
True.
a workflow from 1 to 10, because we cannot, as I say, the patient cannot wait. I mean, again, it's not because only it's the disease itself is very critical and it makes or needs an urgent treatment. It's also because it's evolving. So that maybe the treatment you have for now, it's not the same treatment you have for in 5 months, because the tumor itself has evolved and had developed in other subclones, which they make the disease much more complex. So yeah, this emergency and the fast analysis and so on
Yeah.
is everything really, really alarming and needs an urgent, an urgent research, I would say.
No, that's fantastic. I mean, we know that there's endless use of the AI, as you mentioned, tumor diagnosis, tumor prognosis, for even drug designing, helping the doctor to decide quicker and with much more precision. Um, Now, I think what we're looking for in the future is how to integrate everything in one tool that can make one decision You can make decision of the, the stage of the disease, which drug you give. You can predict the survival. You can have a follow up of the patients. So all of those you can have them in the same tool, but that's require a lot of integration of all those layers of complexities. And AI could help us, but of course that require different tools and how to integrate them. We already talk about the ODC. This is an oncology diagnosis system for imaging, or OPS, like oncology precision system based on molecular aspects, what we call omics, and how to integrate the ODC, which is imaging, to OPC. That require different tools, and that's actually scientists, they are really working hard how to integrate the different methods of the AI to have a precise decision in the end. Yeah, that's
Absolutely.
really
This is
important.
so mainly is to make everything in one network. And this
Yeah,
network is the decisive to make decisions, because otherwise we will not be able to treat just one data and make conclusions. This is a, as you say again, this is a combination of many data and the network between them, which makes that very important. Yes,
yeah,
here we have some real world examples. We know already that, for instance, AI is able to detect breast cancer earlier than radiologists. That does not mean that the radiologists will be eliminated by the AI. However, they can be supported by the AI. Predicting immunotherapy response. We have also some examples of AI-generated drugs for liver cancer in 30 days. Yeah, this is in silico data. Obviously, this is everything could be done very systematic, using the new models and simulating the data and so on. This is nothing we can say this is the end. Obviously, we know that there is the experimental part and also the clinical part. So yes, this is a long going treatment, but it's ongoing. And here I have an example. An example, for instance, of a patient, 45 years old, is a man, and he has a colorectal cancer, which is in stage 3, and how the omics would help us, or with how to make a diagnosis. So mainly, if we have genomics, so we can detect these KRAS mutation. And so, for instance, if we detect the KRAS mutation, we know already that these receptors on the cell, which has EGFR, will not be effective treating them by cetuximab. So here, it's just one hint, for instance. Or for instance, if we have these TP53 mutations, so we know that the tumor is really aggressive, is advanced. So we cannot treat exactly on the same way as we might treat other type of cancer. Transcriptomic, for instance, we have this Wnt pathway. So if we find it already activated, that means that the tumor is already advanced again as before, and this is ongoing proliferation. If we find, for example, in proteomics, this protein PDL1, which is this is important for this checkpoint, and there is an issue there, then again we cannot treat it in the same way. So what can we do? So we know already this. We have already these, let's say, factors or not factors data. Then what kind of action can we take? We can target, for instance, the therapy tailored in KRAS mutant tumors. We can also make some immunotherapy added to this PDL1 expression. And we can also ongoing monitoring via this, how we call that, this ctDNA. This is circulating tumor
circulating
DNA. Yes,
DNA.
circulating
Yeah,
tumor DNA, to detect the
yeah,
minimal residual disease. By the way, the ctDNA, we will talk about this a bit later on the next slides. But we see it's all
yeah,
beneficial, right? Because we can do that in real time. We can reduce the effect. We can be more precise. We can avoid some treatment which could be done in vain, right? Because these treatment would be just
yeah,
expensive and useless, right?
yeah.
What do you think
No,
about that?
that's no, I think it's fantastic. I think AI, of course, will identify those features as you mentioned. we look to the KRAS, the PDL1 for immunology, EGFR for receptors. Of course, all of those features have been hidden in thousands of genes and pathways. Of course, the bioinformaticians with the AI, skills could identify those ones as a key or hub genes that control the alterated pathway in colorectal cancer. And then once you identify those ones, can provide them to the doctors and oncologists and can tell them, look, those ones, they are the most alterated one. Let's treat them and identify them as targets, druggable, key features that you can really target and can modify the disease. This is the way how AI could help us, is to identify the hub genes, the key pathways, and basically target them. And then those examples, they are fantastic, like KRAS mutations for colorectal cancer, and also for immunotherapy using the PDL1. So patients who got the PDL1 highly expressed, they are more eligible for checkpoint inhibitor therapy. So the AI basically could help us. Before, we, as a bioinformatician yourself, you spent hours and hours, or maybe days, to identify those ones. But now AI
Absolutely.
could really identify the hidden ones and then provide them, to use them as a key feature to use them for treatments.
Yeah,
Yeah,
absolutely.
that's
This is
important.
then again the rapid and the fast and the accurate result,
Yeah.
which makes these all these algorithm of deep learning and machine learning very efficient for the future. So here again, maybe this is just a resume of what we were saying before about how these make this revolutionary role of AI in oncology and how we can mix everything together to make a network between non-omics, as we see here, and between the omics and the clinical data too. So basically, it's to detect the hidden relationship. This is what you said already, between different type of data, generating new hypotheses. Then we have to predict the patient response to specific treatment as well as disease progression and survival. And obviously, it's a stratification of the most suitable patient for clinical trial and support in designing a more efficient study. So again, we resume what we said before in this very nice slide. I really like it because it shows how we can how the network between all the data or non-omic, omic, and clinical data could lead to certain output, and from that we can have a very accurate outcome.
Yeah.
Yeah,
So I think if you integrate the omic and non-omic using like multimodal integration between the omic and non-omic, like, there's quite a few examples of AI methods like the ensemble learning for multimodal data. So that could help you to integrate imaging together with molecular data to improve predictive power and precision personalized medicine. I think we have the key example of the immunology. So now you have mentioned already PDL1. Once you identify PDL1, and then of course we have image, then we can integrate both of them and we can provide better, immunotherapy or checkpoint therapy to the patients. Yeah.
absolutely. Yeah,
Yeah.
totally
Yeah. So,
agree. Yeah, there
sense.
is another fascinating topic. I really love it, the single cell. The single cell sequencing is from the most fine and also sophisticated way to use, at least in tumor. Of course, it's not used only on tumor, but at least in tumor, because it is a high resolution genomic and transcriptomic profiling of individual cell, and basically it resolved this issue of this heterogeneity, which is not detectable in the bulk sequencing, right? When we have this bulk sequence, and probably go to the next slide, which probably is much better here, explainable than the other slide. When we see here, basically, when we have a bulk of an analysis, that means a whole genome sequencing, for instance, or whole transcriptomic, we have a broad and average view of the sample. So obviously we see everything, all the subclans, the heterogeneity of everything is mixed all together. We see that from the top, I would say. And it's
Yes.
obviously it's limited to the population level. It's most
Mhm.
economic. Yeah, it's fast, it's useful, and does not detect individual cell. And this is where the role of a single cell is
Sure.
doing. So precisely, it's go more into detail. So it's like
Yeah.
diving into the transcriptomic of the bulk analysis. And yeah,
Yeah.
so basically it revealed this how the cells are heterogeneous, right? Because this is what we say at the beginning. We have a tumor, but the tumor formed this microenvironment, and this tumor itself is going and making so it is dividing obviously, and each cell might so the probability that the cell or the child cell are different from the mother is very high because they are evolving very quickly. They are resistant to harsh environments. They are producing or they are generating a new variant, which are making them stronger and stronger, as we mentioned before. So yes, this single cell really gives this really focus like microscope. So you go and you check directly and dive into the cell to see how these subpopulation are and how complex it is, of course. Yeah, the inconvenient, they are more expensive. They require a lot of analysis because you are analyzing different single cell on the same time to have this overview again about how the tissue is heterogeneous.
Yeah.
How,
Yeah.
what you would
That's
say about the
fantastic.
single cell? I'm finding it very fascinating.
Yeah, I mean, of course, I mean, more depth, better understanding of the disease. Of course, the beauty here, if we are able to integrate both the bulk analysis together with the single cell analysis omics, that give us much better view about what is happening on the disease. Because then with the bulk, you may understand the tumor microenvironment, and with the single cell, you can understand better the interaction between the cells and also the rare subpopulations. Sometimes there's small population that you can miss with the bulk. So by integrating
Absolutely.
both, you understand better the disease. That's why having all those tools will give us better understanding of the disease. As we said in our first session, a better understanding of disease will give us better, or precision on the diagnosis and precision of the treatment. Yeah,
Absolutely. Yeah, completely agree. Yeah,
excellent.
we will finish with these two slides. So we have also special transcriptomic, which is another way also to analyze the tumor. Obviously, it's not analyzed only. This is not for only tumor. It's a technique actually to analyze transcriptomic, but at different level, right? So basically, it's like a Google map in biology. It's like you take
That's
a tissue and you see how the cells are behaving differently inside the same tissue. So this is really also fascinating topic. Basically, I mean, it's just to see how the cells in one tissue behaving differently, and yeah, we can detect these especially regulated gene program. We can check also these tumor microenvironment, the tumor heterogeneity again. And it's a very wide topic again. It helps also to distinguish between human hot and cold zone, basically by this
true.
transcriptomic. It's also
Mm.
detect the immune evasion strategy and detect also therapy resistance, right? So yes, there are a
Yeah.
lot to say in these with this technique. It's a quite novel. I think it's from 2024, 23. It's
Yeah,
not
it's
that.
very new. That's true. It's new. I mean, always, some of scientists they are skeptic to use more fancy techniques, but this is why we have a question why it's matter. I think you already nicely mentioned why it matter. I think knowing the tumor microenvironment, this is the key tool to use a spatial data analysis. Also, cell-cell interactions, very important. And then something else that we forget, I think, is the gradient of hypoxia. You already mentioned inflammation. Also, we can look to the microenvironment, how it changed with the drug response. And we understand also depend on the tumor structure or architecture, what we call, because
Yeah,
basically spatial data is a tissue architecture. It basically can give us the architecture of the tumor. Some of the tumors, they need this type of methods or assay, like glioblastoma, colorectal cancer. They are mainly inflammatory disease, so it's highly heterogeneous. So which means need this type of technique to understand what is happening in the tumor microenvironment. Yeah.
totally agree. And also, this is another point important is signaling. So maybe we can also detect where the signal is coming from, and we can directly
True.
kill the source of this signal. This is really fascinating
Yeah. That's the
topic
future.
and also technique. I think it will really improve for the next years, but yeah, still being skeptic at the beginning could be understandable.
Always.
Yeah, and we will finish our session with the liquid biopsy. So this is another
Mm.
technique, obviously, and this is it's to analyze the circulating tumor DNA from or tumor cells from cells in blood and plasma without a need of surgery or tissue biopsy. This is really interesting because once you have a tumor, obviously there is a rest of the DNA which is just dragged by the blood. So having this detection in the blood, just as I say, the probably very easy, not using this surgery or biopsy, can also lead to big data because you can see already what happens in what kind of rest you have in the blood. So mainly there is another different advantages where I cite here. So minimizing the invasive and repetitive, then enable real-time monitoring, detect minimal residual disease and early relapse. I think it's a really important point. Again, everything could be assembled together to form this big network what we're talking about, right?
Yeah, definitely. I mean, the key point here is not invasive technique. If we can use it to detect, early biomarkers in the blood, that's what we need. And we also can need it to see the progress of the treatment and how patients can respond without, as you said, repetitively taking the samples. So just with the blood, you can understand what is happening. I think circulated DNA is one of the tools, but there are others, the RNA, exosomes, and other that can be used without surgery. I think this is also the future. They are trying to improve these techniques by using very small amounts of or volumes, small volumes, without
Yeah,
taking much more volumes. And using some organ chips or some small devices to analyze the data. Of course, it's all depend on the accuracy of the assay, but also how those biomarkers they are relevant to the disease and stage of the disease in response to treatment. Yeah, thank you.
absolutely, absolutely. I think it's there is a lot to say here, right? I mean,
Yeah.
it's a nonstop. I mean, fascinated topic also, but nonstop thinking and going with the innovation, with the therapy, with how the AI could be involved and which kind of pathway. So yeah, it's a
Yeah.
vast.
If I can add something here on
Yeah,
how AI could be useful
please.
here, because the AI in the beginning will use data coming from invasive, methodologies like surgery, bulk tissue, single cell tissue, having surgery. But in the future, the AI will have already the data coming from invasive, techniques, but will use only non-invasive techniques like blood bio, liquid biopsies, and could have a conclusion predictions. And that actually the beauty of the AI. So there is no need to do surgery, just having this liquid biopsy, and you can build up, precise algorithms that can give you with the precision, the prediction of the stage of this disease, but also the response to treatment and the follow up of the progress of the tumor. So that's actually how AI
Absolutely.
could
And
really help here.
Yeah, and we conclude with that with by the AI. I mean, the objective at the end, it's to minimize the time and to make and to minimize also the analysis, of course, because it's more time is more
Yeah,
money
that's
and more.
true.
Yeah. So at the end, AI will be so helpful. That's the not the treatment, the prediction or the analysis of the data will be very short, and also taking the minimum of the as you say volume or without requiring the surgery or intervention and so on. So yes, the future will be really exciting. So let's hope for the best. So thanks a lot to being here with
Thank you very much. Thank you for
yeah,
this talk.
this
It's really
exciting
exciting.
sessions, and then let's look forward for the third session.
Yes, that's I'm excited about it.
Thank
Thank
you.
you.
Okay,
Thank
bye.
you, everyone. Thank you. I just stopped the recording.